Now, ready?
All right. Good morning and welcome to Aarya Venkat's Thesis Defense. It's indeed my great pleasure to introduce Aarya to you and welcome to his parents too who just joined us. So it's I think a great pleasure for me to introduce Aarya because Aarya is really special in many ways. He started his master's and finished his master's at UC San Diego, working with Michael Hilson in the Department of Chemistry and Biochemistry. And then he started in my lab in 2018 with an interest in protein bioinformatics. And I was truly delighted when he started in my lab because that's the time we were starting this new project on glycosol transferases. And he really took on a really exciting project of investigating the evolution of GPA fold enzymes using a really exciting bioinformatic approach. But since then, he's really expanded onto kinases and more recently, application of deep learning models, which you'll hear from him. And he's also contributed to a variety of collaborative projects. So that's resulted in over 17 co-author publications, and 4 of which he's the primary author. So he's been truly outstanding in terms of his research productivity. So in addition to the research, he's a great team player. He's the first one to go help anyone if anyone needs anything in the lab. He really enjoys mentoring undergraduate students in the lab, and he's a great department citizen. He organizes seminars, and workshops, and he's really organized several symposiums. So I think we're really lucky to have had Aarya with us for the past 5-6 years. I think he'll be really missed, and I think in the past month or so, he's been on the job market and he's got multiple offers, within the short period of time. But then he's really landed an amazing offer from Ginkgo Bioworks. It's a protein engineering company in Boston that's paying him some really attractive salary that we in academia can only dream of. So I think with that, I'll probably give you Aarya and hear what he has to say. Thank you very much.
Thank you so much for that introduction, Dr. Pennon. So, hi everyone. You know who I am now. I'm going to be talking about modular evolution in proteins, and this is actually very relevant to protein engineering. And specifically, as Dr. Pennon had mentioned, I'm looking at kinases and glycosyl transferases. And so, when we think about modularity, what actually is it? It's the idea that complex systems can be broken down into simpler pieces. Just like in the human body, you can break down organs from an organ system into different organs like the brain, lungs, spleen, so on and so forth. Just like within the cell, you can break it down into components like the nucleus, the lysosome, ribosomes, the ER, so on and so forth. You can do the same thing with proteins. Proteins can be broken down into domains, different domains that act together in tandem to form an assembly that has some specific function. And you can also break down proteins into individual pieces that have assembled over evolutionary time to compose the proteins we see today. And so when I talk about proteins, really, we should reflect on this engineering or systems biology classification of proteins. That is, we can talk about them in the forms of 3 classes: readers, which recognize modifications that proteins add; writers, which actually add the modifications; or erasers, which remove those modifications. And I'm going to be talking about, as I mentioned before, 2 writers, kinases and glycosyl transferases, which are really transferases by any other name. What do? They essentially take some nucleotide phosphate substrate and they extract a piece of it. That is, in sense of kinases, a phosphate from ATP, or in glycosyl transferases, a sugar from an NDP sugar. And when we have, pulling back a little bit, proteins use these modifications for language, but cells actually use sequence as their own specific language. So in the central dogma of biology, right, you have DNA sequence that gets converted into RNA sequence, and that gets converted into a protein sequence, right? Cells understand this complex code to perform a vast diversity of functions. And recently, my latest work, which I'll talk about at the end, is using AI to help understand this language. And so artificial intelligence, we already know, has understood and comprehended grammar and syntax of regular language. We can see that with ChatGPT, we can see that with Google Translate. When you write in your phone, you have predicted text, right? The quick brown fox jumps over the lazy dog. Why can't we apply the same thing to sequence? We take a protein sequence and we could mask out a piece and see if we can figure out how to integrate that back in. And in fact, in the last couple of years, that's exactly what's been done. And recently, there's a massive protein language model, is what we call them, that has been developed that essentially does this. What it does is it takes a sequence, it translates it, Google Translate, right, to a language that a computer can understand. What is that language? Numbers. And with numbers, computers can do various sorts of operations. They can multiply, they can perform logistic regressions, so on and so forth, all to predict information about structure, fold, evolution, and function. And so in my last talk, after I talk about the first two chapters, I'm going to talk about how to use these protein language models fundamentally to predict glycosyltransferase function. So I'll jump into my first chapter here. I've given this talk a lot, and most of you have seen this a lot, so I'm going to keep it brief. But glycosyltransferases, as I mentioned before, they're writers. What do they write? Well, they write letters, these monosaccharides, mannose, glucose, galactose, so on and so forth. And they form words. Those words could be larger sugars, oligosaccharides, or those words could be proteins, RNA, lipids, that they attach some sort of monosaccharide onto to perform various signaling events, controlling the unfolded protein response, controlling cell fate, so on and so forth. And how did they evolve to write these, how did they evolve this writing function over time? We previously published a paper back in 2020 to essentially look broadly at the phylogeny of these glycosyltransferases across the tree of life. And one thing we noted, glycosyltransferases, we can break them down into inverting and retaining enzymes, whether they invert or retain the stereochemistry of the transferred sugar. And we find that inverting and retaining glycosyltransferases occur together in multiple areas across the tree of life, suggesting that these mechanisms evolved independently multiple different times. To do so, you must have some sort of intrinsic modularity, some way of quickly swapping out different pieces together to be able to switch function like this. And so we wanted to investigate this further, and that's what formed essentially my first thesis chapter. And so we, over 700,000 GTA sequences, we essentially looked at evolutionary conservation, and specifically, we looked at the most conserved element in these glycosyltransferases and in proteins in general. That is the hydrophobic core. The hydrophobic core is a conserved, typically thought of as static element that is seen in all proteins and is what the protein folds around. And we see that in this hydrophobic core, we see in terms of conservation, it seems to align in three separate pieces. Here we call the Rosman fold area, given that this is a sequence region that corresponds with a lot of Rosman fold enzymes. Here we call the most conserved region, and we relabel it later, but I'm not going to introduce that title just yet. But we call it the most conserved region because it has the highest conservation. That's what the y-axis is, its level of conservation. And then here is something that's GTA family specific, only found within fold A glycosyltransferases and quite variant within. These three different modules essentially represent how the glycosyltransferase progressively evolved over evolutionary time. This first piece that I mentioned, that is this very conserved region, is what we call the phosphate binding cassette. This is a region that was first discovered by Daniel Topic's lab in the University of Tel Aviv. And essentially what he found was there's a conserved region between, between Rosman fold enzymes and a variety of other enzymes that fundamentally forms this beta, alpha, beta segment. And we were curious, is that beta alpha beta segment fundamentally conserved within GTAs, which are also a Rosman-like family? And we did find through their sequence analyses and through a combination of sequence and structural analyses that yes, GTAs indeed have their own phosphate binding cassette with a miniature hydrophobic core. Over evolutionary time, we then extend this piece with an end lobe, something that's conserved across all Rosman folds. This Rosman end lobe, noted by this beta alpha repetition, is essentially helpful for binding nucleotide diphosphates, but is also helpful through this hydrophobic core for adding a massive amount of stability towards the enzyme function. And so that was what we hypothesized as being the contribution of this modular segment. Finally, the last piece in this evolutionary history of this enzyme is what we call the C-lobe tether. What's important about this is that essentially it tethers the catalytic base of these glycosyl transferases to this phosphate binding cassette. And so it's present right behind the active site of these enzymes. And we thought, hey, maybe we see it varying across these different glycosyl transferase families. Maybe it's involved in possibly regulating catalysis in some way, especially because one of the residues is only 3 residues away from the catalytic base. So we wanted to investigate that. First, we performed this sort of sequence analysis, looking at inverting and retaining enzymes, and we found just measuring the distance of these 2 residues right here, that in fact there is a key difference between retaining enzymes and inverting enzymes. Inverting enzymes generally tend to have a hydrophobic contact between 4 to 7 angstroms. Retaining enzymes have a hydrophobic contact anywhere between 4 all the way to 13, 15 angstroms. So in some instances, there's no contact at all because the average contact is between 4 to 7 angstroms, suggesting there are actually packing defects in many of these retaining enzymes. And so we were curious, okay, what's going on? Why are retaining enzymes having these packing defects? Why are there core packing changes? One thing we noticed is that many of these retaining GTs that have a distance, a hydrophobic contact between 4 to 7, suggesting they are contiguous hydrophobic core, they're all related to GT2 enzymes, inverting enzymes. And these enzymes are some of the most oldest in terms of evolutionary conservation across glycosyl transferases. And what that suggests is that maybe some of these GTs possibly evolved from these GT2s, these retaining GTs, and so they have a more conserved hydrophobic core. Nevertheless, many of these retaining GTs seem to have, over evolutionary time, evolved multiple ways to essentially coordinate catalysis. And what's important to remember about this is that retaining GTs actually do not use the catalytic base during catalysis. Instead, they use the UDP donor itself to deprotonate the acceptor, allowing catalysis to occur. So it's possible that these core packing defects are, one, a result of these the loss of this XCD constraint, and 2, the result of these enzymes being able to really sample a variety of different possible functions for acceptor diversity, for substrate specificity, and so on and so forth. And so up until this moment, I've been doing mostly computational analyses. You guys know that I'm a computational biologist, so my mouse model is next to a keyboard. I was hoping for a louder laugh, but it's all right. But I was curious, what happens when we actually, let's do some experiments. Let's actually mutate this hydrophobic core and see what happens. So I did this first in silico, and we noticed that there were, in fact, we use B3GNT2, which is which is one of the key enzymes that the Mormon lab uses. And we noticed that there's actually a difference, or there is motion of this XED motif in what we found 2 different conformations, an in conformation and an out conformation, depending on whether this XED motif swings in and coordinates with this water, which then coordinates with this hydrophobic core. What's interesting about B3GNT2 is that instead of a hydrophobic residue in its core, it's actually got a 3 polar residue. And so we're curious, what satisfies that contact? Why does it have it in the first place? What's going on generally? And I thought, okay, so it has these two different conformations that it's swinging back and forth for. When it swings inwards, it's forming this hydrogen bonding interaction with this threonine. What happens if we abrogate that interaction with this water? What if we mutate the threonine to a hydrophobic residue, knock that out? What's going to happen to catalytic activity? Daniel Tirani from Dr. Mormon's lab kindly performed experimental and kinetic analyses for these. And we found that upon donor saturation, we're essentially getting a 2.5-fold increase in kinetic efficiency between the wild type and this T336I mutated into isoleucine. A 2.5-fold increase in kinetic efficiency, essentially where the Kcat doubles and there's a small improvement to Km. And so what we're thinking is that by mutating, by abrogating this water interaction, by mutating this threonine, this XED motif no longer has any favorability to swing into this hydrophobic patch, and now it spends more time in this out conformation. And if the donor and acceptor are there, it's more likely to deprotonate the acceptor, resulting in increased catalysis. And so that was our reasoning for why there might be an increase in kinetic efficiency. And it's really interesting because these small variations over time contribute to massive functions. Starting from this phosphate binding module, we add this addition of this Rosman fold, we add this addition of the C-lobe tether, the XED and C-his as well, that contribute massively to the function of these glycosyltransferases. And then, for modern GTs to exist, we've added a series of hypervariable regions that are not conserved at all across different glycosyltransferases, only conserved within families. And they have a lot of family-specific functions we've noted, and things like acceptor substrate specificity, donor substrate specificity, binding affinity, and helping coordinate the catalytic site. That has, and these different modules have essentially, over evolutionary time, helped tune and essentially engineer these GTs to become the diverse language or linguists that they have become today. And so it's sort of like the story of Goldilocks, right? Goldilocks says, you know, this bed is just right. Forgive me, I haven't read Goldilocks in a long time. But in essence, this phosphate binding cassette, this C-lobe tether, this N-lobe module, have all really worked hard to essentially bind this nucleotide diphosphate sugar. And that makes sense. In terms of the number of donors available for these enzymes, there are only like 10 or 11 for metazoans as a whole. In terms of acceptors, you have an innumerous amount. You have multiple different proteins, multiple different sugars, multiple different lipids, and now we even know RNA is a potential substrate of glycosyltransferases. And so acceptors are sort of an afterthought in this evolutionary trajectory. Instead, most of this enzyme has these conserved modules that have specifically bind this UDP sugar with small differences that might influence what sugar they actually bind, with hypervariable regions instead focusing on the actual acceptor that's bound. And so it seems that the evolution of these modules have sort of guided the kinetic cycle of these enzymes. If you know anything about how GTAs function, GTAs typically bind a metal cation first, then they bind the donor substrate, then they bind the acceptor. And so it couldn't be any other way, because fundamentally the acceptor, if it bound first, it would essentially prevent or negate the binding of the donor. And so in this way, we believe that the story, the evolutionary story of these glycosyltransferases essentially guided this kinetic cycle. So I'm going to talk a little bit about shifting very far away from glycosyltransferases into kinases, a work I did just last year. And I want to talk about, in the same frame of modularity, how exactly these kinases end up modulating their function. And what's interesting about kinases, something I wanted to mention, because when I saw this, I became incredibly envious of everyone in my lab that works on kinases. I aligned 800,000 kinase sequences, and then I built a sequence logo. And if you're familiar with sequence logos, this, for 800,000 sequences, is incredibly well conserved. If you tried this for glycosyltransferases, which I have, it's basically an empty desert. Everything looks like this region all across the board, except for a few key pockets. And so I saw this and I was like, okay, they are, when people say they're conserved, they are really well conserved. And so I'm curious, where does modularity actually come into play? How do these kinases actually differentiate themselves in terms of function? And we published, Wayland Young in our lab, published a really great story on the evolution of diversity in the holozoan tyrosine kinome. And what he noticed is that many of these tyrosine kinases essentially have variations in the N-terminal flanking segments. They can have multiple different types of variable domains, some that contribute to intracellular functions, some that contribute to extracellular functions, some that contribute to immune cell signaling. And we know that these different functions can also be found at the C-terminal end of these proteins. So the domain itself is super well conserved with maybe small mutations that fundamentally aid in tuning that function, but the bulk of the work seem to be done by these flanking regions that essentially help modulate the kinase function. And so we wanted to focus more on these C-terminal regions. And in particular, we noticed that these double cortin-like kinases, DCLKs, that are involved in, that are expressed in brain tissue and actually involved in a variety of things from microtubule binding to microtubule migration, and the development of neuronal cells, development of neurons. We wanted to investigate these because what's interesting about these enzymes is that they have multiple different isoforms, and specifically, the only variable segment between these isoforms is the C-terminal tail that contributes into different variations of function. That is, one is this DCLK 1.1 constitutively active, DCLK 1.2 constitutively inhibited. Complete opposite functions. And we're curious, what is the mechanism behind this? Why exactly are they operating so differently? DCLKs have 3 different paralogs. I'm only going to focus on DCLK1 because that's where we have the most information, but we are continuing on looking at the other paralogs of DCLK. So I wanted to look at the mechanism of these. So just to show exactly what these look like in 3-dimensional structure, many of these are AlphaFold models. This is the only actual crystal structure that we have. The only reason we have a crystal structure is because this one is actually constitutively auto-inhibited. So these enzymes are both auto-inhibited. These enzymes are both active. And essentially what we see is that these, there are 4 isoforms, 2 which don't have these DCX domains that actually bind the microtubules, 2 which do have the DCX domains, and 2 that are active, 2 that are completely inhibited. And we're curious, okay, why do you have 4 different isoforms? They're almost completely identical. We're only going to be focusing on the ones without DCX domains because that's actually what we were able to experimentally work with, because these DCX domains are really hard to purify. But we're curious, you know, what makes them tick? Why is one super active? Why is one super inhibited? So we did first an evolutionary analysis of these DCLKs. And what we noticed is that over evolutionary time, 1, we see the increase in the number of DCX N-terminal DCX domains over evolutionary time. That means as you go into vertebrates, as you go from invertebrates to vertebrates, you get more DCX domains. We also notice at the same time a correlated increase with tail length, the C-terminal tail, as you go from basically these very ancient proteins or ancient animals, or I'm not sure, ancient cells, all the way to vertebrate cells in modern sort of sequences. And we noticed an increase in C-terminal tail length. And we're curious, what is the function of that? Why exactly are they increasing? It's in these vertebrates that we also notice duplication event. We have DCLK1 and DCLK2, which are very closely related, but they duplicate at some point during the emergence of vertebrates. So you essentially have 2 copies of very similar enzymes that still seem to have different functions. And that's something we're also trying to investigate in parallel. But focusing again on DCLK1, we looked through UniProt and we noticed that, in fact, in a lot of mammals, multiple mammals have multiple copies or multiple isoforms of these enzymes. And in fact, this is something that struck me, is that in UniProt, there is an inadequate amount of isoform data. When you actually look through Uniprot, there's very little known. Most people, when they look at isoforms, they're like, okay, this maybe it's a sequencing mistake, or maybe they toss it out or they truncate too early. And so you have a lot of isoforms that are really missing throughout life. But these are really important because we're seeing more and more that these isoforms are fundamental to the function of these enzymes. So we wanted to hone in on why exactly you have such a massive enrichment in isoforms across mammals. So let's take a genomic view. Everything or genomic view of these enzymes. These enzymes have 17 exons. Everything before exon 15 is identical. So it's only after exon 15 that you have variations amongst DCLK 1.1 and 1.2. 1.1 is active, 1.2 is inhibited. 1.1 has an extra exon, exon 16, that is not seen in DCLK 1.2. And so this extra exon, exon 16, is then followed by what's called a phase 2 intron. What's a phase 2 intron? A phase 2 intron is simply an interruption between the second and third codon that results in a frame shift of the codon. So both of these have exon 17, but this is a different exon 17 to a frame shift than this exon right here. So when I show the actual what this looks like in terms of protein structure, this shift in reading frame of DCLK 1.1 results in this following sequence: LDH, GFT, so on and so forth. This region before VIA being all the same identical sequence. Here you have this region TTA that it starts off. And then there's actually quite a lot of related, interestingly enough, sequence conservation across. But you have a couple of residues that are different that seem to contribute to differences in their function. So this difference in frame shift, you might be wondering, why is this important? I'm not even sure why we're interested in this detail. Well, the reason we're interested is because when we actually look at the molecular or the proteomic level, we notice that through Bayesian sequence analyses, there's a lot of patterns that are specifically conserved in DCLKs that are not conserved in other kinases. Essentially, what we've done here is we're comparing DCLKs versus other kinases and trying to pull apart what patterns are unique to DCLKs, what patterns are completely differential across kinases. And we notice that, and this is really important for protein engineering as well, is that we have a couple different residues that we can really look at that might trigger particular functions in this enzyme. And if we work with those, if we mutate those, perhaps we can induce alterations of function. A second thing, I mentioned that TTA region that was different between DCLK 1.1, DCLK 1.2. That TTA region, that second threonine, is actually phosphorylated. So that's a difference that's seen in DCLK 1.2, which is constitutively autoinhibited. That's not seen in DCLK 1.1. So okay, I'm done with sequence. What's the actual structural basis and what is going on in terms of structure in this detail? So this is the average kinase, a really well-known kinase, protein kinase A, and this is what it looks like. You've got this region right here. This is the hydrophobic core in green. This is a allosteric hydrophobic core. I'm not going to explain too much about the hydrophobic core, but if you have any questions, I can explain it more in detail. But this, what we call the C-spine, this green hydrophobic core, has this little open cavity that ATP can essentially enter and complete that C-spine. In essence, what that does is it one, bumps up the stability of the enzyme, bumps up the thermostability of the enzyme, and it essentially completes the C-spine, and this allows for stability of the ATP to then transfer a particular phosphate. What we noticed in the C-tail of DCLK 1.2 that's constitutively autoinhibited is that this region of the C-tail, the region that was highlighted by our Bayesian statistical analyses, seems to form this hydrophobic patch that actually integrates itself within the C-spine of this hydrophobic core. suggesting that it might act as a type 1-like inhibitor of these DCLKs. Additionally, that threonine I was talking about that gets phosphorylated, just like ATP has a phosphate right here, we essentially have a phosphothreonine that mimics the gamma phosphate of the ATP that binds to the G loop, just like in the same mechanistic way as regular kinase. And so what we have here is essentially an alternative splice variant that now mimics the structure of ATP that has entered the pocket and has constitutively inhibited this protein kinase. This region right here is missing in DCLK 1.1, which is constitutively active. And what we think is that by missing this key ingredient of this ATP-like structure, you're going to have a lot less stability. Maybe this gets knocked out of the pocket, and you have, regular catalytic activity again. So let's actually experimentally look at this. What effects do these different isoforms have? So here we're comparing DCLK 1.1, shown in red here, with DCLK 1.2, shown in blue here. Active, inactive. And we see that the inactive version, where the tail is actually bound and forms that ATP-like structure, is about 16 degrees in thermostability increase compared to the active-like structure. If you had kinase bound to ATP, we'd see essentially the same thing, an increase in thermostability, suggesting that when we're experimentally dealing with these DCLKs, this tail seems to actually be constitutively bound to the kinase core. When we just cut off the tail entirely, we have about the same level of stability that we have with DCLK1. So it's about the same level of thermostability. So they're all both constitutively active, and they're maintaining sort of the same level of thermostability. And what that suggests is that the tail experimentally or in vitro is not actually docked to the kinase domain here. When we compare that cut tail to the constitutively inhibited region, we're seeing that same 16.5 degree increase as compared to this cut tail. And so what we're thinking, and let me clarify some of the terms here, this IBS region is where the tail, that ATP-like region, is actually docking. And then we have an intrinsically disordered segment that we thought might add an additional set of functions to this kinase domain, that we haven't fully interrogated what that IBS region does. So taking that thermostability into account, we want to get, yes.
That was calorimetry?
Yes.
Oh,
Oh, I'm sorry. I should have said that.
it's reversible.
Yes. Okay. And okay, so you have let's look into the dynamics perspective of isoform-specific thermostability. So we ran molecular dynamic simulations, and very simply, what we're interested in seeing is how did these tails actually behave in silico? And what we notice is that, one, at the top right here is DCLK 1.1, the active version, and at the bottom here is DCLK 1.2, the constitutively inactive, the inhibited, auto-inhibited version. And this auto-inhibited version is really tightly bound and forms this helical pocket, which is shown by this red, this ATP region, shown in red right here, versus the DCLK 1.1 has no helical region. These blue are beta sheets, has no helical region whatsoever. So it's just a loop essentially in dynamics. And so what we're seeing, our dynamics are basically showing that this region of this IBS is very, very dynamic. It's not maintaining sort of the helical loop that we would expect. And it seems to be sort of pulling out over time, over dynamics time, out of this pocket. So in general, we have significantly less stability of the tail within this pocket than we do with DCLK 1.2, which stays in that pocket really tightly. And the reason for that seems to be these small variations that occur after exon 15. So as I mentioned earlier, we did these Bayesian analysis and we highlighted a couple different residues over this enzyme that we thought maybe if we mutate, we could essentially restore kinetic activity of DCLK 1.2, the auto-inhibited version. So we targeted a few of these residues, D398, N400, some of these hydrophobic residues. And we expected, okay, can we actually knock that tail out of the pocket and restore some of the stability? And we found that if you do knock out, or if you do, mutate some of these residues, D398 to alanine, N400 to alanine, V682 and 684 to threonine, and D691 to alanine, we do notice differences in thermostability. Some of these, especially in these G-loop regions, seem to remain the same in terms of thermostability. But if you knock out the actual tail itself, or if you, keep using the word knockout, if you mutate the tail specific residues yourself, you get a very similar thermostability to DCLK1 catalytic domain only, that is with the truncated tail. And so we have about the same level of thermostability just targeting these mutations alone. This is a kinase assay, so we're looking at phosphate incorporation over time. And essentially, our catalytic domain kinase only has the highest phospho incorporation. DCLK1 is not as high, but we have the tail there, and so perhaps the tail is playing some way in regulating the amount of phosphate incorporation. You have almost no phosphate incorporation for DCLK 1.2, the auto-inhibited, as expected. Then these mutations that we perform, specifically N400, or sorry, D398, which is located right here in the G loop, and also some of these tail-specific mutants seem to restore the actual kinetic efficiency of this enzyme. So D398 actually restores it almost to the level of the KCAT enzyme, and then these other mutations of the tail itself restore it very similarly to DCLK 1.1, suggesting that essentially they're knocking out the tail and returning the kinetic efficiency to what we would expect with DCLK 1.1. So I've talked only a little about this. This paper was published last week. So if you're interested in more of the functions that we've uncovered, what we call this tail is an evolutionary Swiss Army knife. It's not just got this one function where it mimics ATP, but it also mimics the actual kinase protein substrate that is bound, has this intrinsically disordered segment that we're still trying to find more about. It has a variety of other segments that are conserved between DCLK 1.1 and 1.2 regions before exon 15 that contribute to what we think is differences in regulation of the enzyme. And so this really modular and interesting evolutionary Swiss Army knife, we're continuing on to investigate with members of my lab, like Grace, who are really digging in and figuring out, what these other parts of the tail actually do. Okay, so last part, my work in deep learning, trying to look more at glycosyltransferases. So sort of returning back to this, these glycosyltransferases. One of the key issues with glycosyltransferases is that for predicting donor function, we know that some of these glycosyltransferase donors can be tens of thousands of dollars for very little amounts. And so ideally, if we had a tool that essentially allowed us to hypothesize what donor a glycosyltransferase sequence might find, perhaps that would help cut down for experimentalists on the time and cost people have to spend to actually try to identify these different sugars. So going back to my original model on protein language models, essentially all we're doing is taking a sequence, we're translating it to a series of numbers that computers can understand, numbers that essentially computers can then use to compute, complex mathematical formulas to do a variety of things like prediction of these enzymes. And so one particular protein language in general, or in specific, is called ESM2. This was produced by Facebook last year. And essentially what they did was they took Uniprot, about 68 million unique sequences, and they crafted a deep learning model that really learned the language, the grammar, and syntax of these protein sequences. And they did it in a method called unsupervised learning. What is unsupervised learning? Very simply, it's giving the raw protein sequences to a deep learning or neural network, and the neural network comes to its own terms of what these features actually mean. And it can find features like evolution, structure, chemistry, different protein functions, and other protein relationships simply by just looking at these 68 million sequences and just trying to uncover what the patterns are. And it can do that because it's just comparative numbers. And computers are very good with numbers. So just a quick overview of what deep learning is. I kept mentioning it. Very simply, you give an input to a model, you get a prediction. So what is the model actually doing? It's learning to fit data. You have multiple data points. These might be protein sequences. These might be features about protein sequences, like their structure or maybe particular residues of interest. And all it's doing is plotting some sort of function, in this case, a logistic regression, on the data and trying to figure out how do I optimize this logistic regression to fit the data the best I can. And once it fits that data, you might have a really good prediction on your hand. And so it's teaching itself essentially how to best fit the data. And we've used these machine learning and deep learning models for a variety of different purposes. For post-translational modification prediction, led by Zhongling Zhao for a program called Phosphormer that predicts protein kinase-specific phosphorylation, knowledge graph pathway prediction, evolutionary tree generation, and estimation of sequence conservation, led by Wayland Young. And then my mentor, Rahil Tajale, who did full prediction for glycosyl transferases and function prediction, very early function prediction using classical machine learning for predicting donors. And building on all of this, I wanted to create a donor classification model that everyone could possibly use, just to see, or just to create an easy tool for everyone to, get a good hypothesis about what donors a glycosyl transferase might bind. And so here, I'm going to quickly give a live tool demonstration. Live tool demonstrations always work out perfectly, I've heard. And so what this tool is, very simply, you can take a protein sequence, and so I'm highlighting here an example protein sequence, and maybe am I really? Oh no. Okay, hold on. How do I okay, let me try this.
That's good.
Yeah, I'm trying to escape. Dear God. This would happen. Like I can literally, I have it right here on screen. Can I just show it like that? Okay, thank God. All right, so I have a sequence right here, and I'm going to copy as best I can. Here's the protein sequence, and I hit predict, and that's it. It just takes in a FASTA sequence, and what it's going to give you is essentially prediction. So this is a GT31 family. It's closely related to manic fringe, I think. It's predicted to bind galactose. And so this is actually a galactosyl transferase, so that's correct. It will also give you information about the GT. It'll link to KZ. It'll tell you what mechanism it is, what plate it is based on our phylogenetic tree. It'll also tell you how confident it is in that prediction. So GT31, it's pretty confident, about 95% or so. And for galactose, it's about 80% confident about that prediction. And that's because my model in general is about 88, 89% confident for the actual donor prediction. So it'll tell you there are essentially two models here, one that predicts what family it belongs to, one that predicts what donor it belongs to. All right. And now I have to return. Okay, I'm back. Okay. Additionally, what I will talk about is sort of an explainable component that I've included into this model. And I'll do that in just a bit, but let me first explain my workflow here. First, we take UniProt data, and I took about 55,000 sequences for our donor model. And essentially, these are all manually curated. So that is, I specifically looked for sequences that were very diverse across the glycosyltransferase sphere, that were another thing, well annotated in the UniProt database. If it's poorly annotated, if it has a star under one I didn't include it. So I only included sequences that had at least one other step of verification. So someone had actually looked at it and said, okay, this is actually a sequence that does this activity. I took that sequence, those sequences, 55,000, split them into training, validation, and testing sets. And these training, validation, testing sets are essentially different ways to ensure that my model is actually performing the way I intended to do. I go and actually do the training. So I'm taking this protein language model ESM2 that was trained on millions and millions of protein sequences. And ESM2 is essentially already knows the general the grammar and syntax of these protein sequences. All I'm doing is teaching it what a glycosyltransferase is. And that way it can better predict on glycosyltransferases. And then I take that classifier, I can put it into this Glydentify platform, and then we have our classifier. So it doesn't have to be just donors, it doesn't have to be just glycosyltransferase family, it can be any component about glycosyltransferases. And so this is a way of sort of democratizing these AI tools for everyone to use. You know, you can build your own software, you could include it in Glydentify, and you'd be able to essentially, have that out there and say, hey, we're predicting X about these glycosyltransferases. And so you might ask, why fine-tune the ESM2 model? Doesn't ESM2 already know all these features about protein grammar and sequence? Well, the reason is it's trained over 68 million sequences, right? It doesn't, just like, if you're taking the entire population of the United States, 310 million, last I checked, you don't know anything specific about Georgians. You don't know anything specific about Californians. You just know things about the United States as a whole. And so really, when we talk about fine-tuning deep learning models, what we're doing is essentially teaching it what is a Georgian in the United States? What is a Tennessean? What is a Floridian? I don't even know if I'm getting those names right, but hopefully I am. But we're essentially training ESM2 to be more specific. We're using that. We're essentially converting these sequences into what are called embedding vectors, these arrays of numbers that are higher dimensional representations. And then we're doing math. We're doing logistic regression. Over and over, we're just plotting this function, seeing if we hit the data correctly. Does the data actually match what we know to be true? And it's using a function called cross-entropy loss that is very simply just a way of teaching this deep learning tool. Is it actually on point for guessing, or is it going the wrong way? It's like an energy minimization. You only have two directions. You can go up, which is bad for energy minimization, or you can go down, which is good for energy minimization. And so it's using that to essentially figure out, is it actually going in the right direction for prediction of these enzymes or prediction of these functions? Once we get the optimal plot, we can just take the output of this data, this logistic regression, what are called logits, and then we can get a probability, 80% galactose or 80% glucose, 30% galactose, 20% mannose for whatever sequence. And so this donor classification performance, I've gotten about 88, 89% actually now. And we're able to actually split that across different donor sugars. So I can see how is N-acetyl galactosamine performing versus glucose, versus galactose, versus glucose. And that will actually tell me, how do I better refine my data set? Maybe if you look down that here, 78%, is only guessing at 78%, that tells me that maybe my data set isn't as, well balanced as I should have it. And so I can go back and help improve the data set for GalNAc and get a better accuracy for these enzymes. We are comparing it against our previous model, this eLife paper we made in 2020. And what's important to know is that while we're getting about the same level of accuracy, there's a difference in the number of sequences we've really built our data set on. Machine learning is what was used for this original eLife data set. And machine learning means that you're hard coding certain features. This is called feature engineering. And these hard coded features are sort of strict. It tells you, hey, for this set of 713 sequences, these are the features we found most important. But in reality, protein sequences are significantly more flexible than that. So we use a deep learning model to sort of help alleviate some of that flexibility or deal with some of that flexibility. That means that we have to deal with a lot more sequences, but at the same time, we can get a model that is more robust and more generalizable no matter what sequences are out there. So while we're getting about the same accuracy, what really matters is generalizability. And that's what we're really aiming for here. So my final piece of this is adding an explainable component. And this is really going to help sort of the protein engineering side of this project. So given a galactosyl transferase, for example, this is B13 galactosyl transferase, one of the ABO blood group glycosyl transferases. What I've done is essentially include a feature that allows you to mask out different parts of this protein. Essentially, given this massive sequence here of 340 amino acids, turn the first 10 residues into X's. That means that they're going to be ignored by the model. Turn the second 10 residues into X's. It's going to be ignored by the model. You plot that into a heat map that says, hey, what did it predict? Is it a GalNAc? Is it GlucNAc? Is it Gal? So on and so forth. And just see if there are variations in the prediction. And if there are variations in the prediction, that tells you two things. One, it tells you what are the most important components for the actual prediction itself, and what are the regions that are important for if you were to predict a different donor, so GlucNAc versus Gal. And what that might also tell you is how do we actually engineer this protein? What are the specific residues that we can pick out using both this and biochemical intuition to better understand what residues we need to fundamentally mutate to contribute to donor promiscuity or change in a donor. And so what I found with this enzyme is fundamentally, it's really highlighting that phosphate binding cassette that I was mentioning at the beginning, which makes a lot of sense given that small mutations in this region could contribute to alterations in activity. It's also highlighting some of these other regions that are containing the residues that actually do get mutated, depending on whether you're A blood type or B blood type. It's not fully figuring out some of these hypervariable regions that some of these residues are on. But it is, I think with further refinement of the model, we'll slowly be able to get there and be able to figure out, at a more fine-grained residue level, what are the residues that are actually contributing to particular donor specificities. So some of the challenges I've had: overrepresentation by certain families. If you look across the UniProt database, GT2 families, cellulose synthases tend to be heavily represented. And part of that is just, what is easy to experimentally analyze. Some GTs are significantly easier to purify than others. So it's really important to balance your data set. How do you, these are sort of more deep learning focused, is like, how do you train your model appropriately? How do you prevent your model from being so constrained within the data set that it learned off of? How do you make sure that it actually generalizes well to unknown sequences? And then finally, you can see I've only done it on 5 donors, but obviously that's not all the donors that are out there, but they are the most well-represented donors. Gluc, Gal, Man, GlucNAc, GalNAc, you could easily find 10,000, 15,000 sequences, well-represented sequences for each of these. Whereas some of these, like Xylose, I can only find maybe 2,000 sequences. GTAs that specifically bind silos. And maybe that's not enough to build a deep learning set. Some of these I can only find maybe 600. And those are sequences that, are well annotated on Uniprot. So I am limiting myself, but I'm trying to build a very robust data set. And so it's sort of a constant battle. You have to know your data set really well to build a really good model. So in conclusion, I know it's been a long talk and I know everyone's hungry too. But just a quick conclusion, we've modularly adapted this ancestral core of full A glycosyl transferases and understood generally how they've evolved these diverse catalytic and substrate binding functions. We have understood how variations in the flexibles tail segment of DCLKs show how they've really adopted these modular changes for specialized regulation. And this really paves the way for innovative kinase targeting strategies. For example, you remove the tail, could you then use that tail or an engineered version of that tail to be an inhibitor for other kinases? Finally, we developed a user-friendly tool that essentially leverages these novel protein language models to classify and predict GT functions with a relatively high accuracy. With an explainable component that highlights the significance of these residues, hopefully, this will democratize the availability of these AI tools for protein analysis and engineering for everyone. With that, I'd like to leave you on a final note. That is, if people have spoken to you about, oh, we're undergoing an AI revolution, you should really know that the AI revolution was like 7-8 years ago. Now everything is AI. Every single day you're using AI. Every time you text someone, every time you email someone, every time you Google search, every time you're shown an advertisement, that's all made by AI. AI is already here. We're in the middle of AI, or we're at the beginning of AI evolution. And like organic evolution, it's modular. It's never going to be one massive AI tool that does everything, not for another 30, 40 years. But instead, it's people like you that have the expertise of domain level knowledge of your specific enzymes that can create AI tools. They're becoming easier and easier to understand and use. And you can really use them to fundamentally create new tools that do these prediction and classification tasks. And so, new grad students that are here, honestly, make use of these tools. I only started learning about deep learning and AI seriously over at the beginning of the summer. So I genuinely think that it's going to become easier and more available for everyone to use. With that, I would like to acknowledge a lot of people. Grace Watterson in my lab, especially, she's helped me so much, my undergrad. Nathan and Brady and Zhong Liang, who taught me so much about AI and about protein kinases. Safal, Sammy, who taught me so much about just general biochemistry and experimental work. So many undergrads that we've had before, really brilliant. Niril, Claire, Ishan, Sean Gill helped me originally setting up this Glidentify model. Kat helped me with a lot of different stuff. Waylon, Rahel, who taught me the essence of how to design figures and how to, and glycobiology in general. Our collaborators, Pat and Claire Ayers from the University of Liverpool, Dr. Mormon, who helped with our first chapter here, my postdoc Freya, obviously, and then my committee, Dr. Cunnan, Dr. Mormon, Dr. Haltiwanger, and Dr. Woods. BCMB staff, Angie, April, Sandra, and all the advice over the years from Zach, Bree, Aaron, Chris West, and so many others. Special thanks to my parents and my sister at the back here. And then I know we're supposed to have typically funny stuff. Oh man, the GIF is slow. Okay, so here we went to Blood Mountain, me, Laju, and a couple others. And I'm known for being very clumsy, and it was a really hard raining day. I was like, I'm not gonna slip. I'm okay. Immediately slipped, and Waylon created this nice frame-by-frame analysis of this. Here, Liju, over the pandemic, we were making samosas, and I didn't have a rolling pin, and Megna gave me this wine bottle, and that was what we had to use to actually roll the actual, bread for making samosas. my dog Freya, who is here with my sisters and my cousins, just having a ton of time. Here's her at 1:30 a.m. in the morning, just looking at me. I don't know why. And then just all the fun I've had with friends over the years. And with that, I'll leave it to you.
Questions for Aarya?
Yes.
So you mentioned at the end that, that there were only 5 donor sugars that were used to train the model. So does it predict outside of those 5?
That's a great question. So no, it doesn't. It'll predict sequences. So let's say that you have, for example, gout1, gout2, right? Those sequences, it's going to try to constrain it within the 5. However, I'm building currently an other section for sequences like gout, for example, that are, binding stuff like galacturonic acid, all glucuronic acid, so on and so forth. And making sort of an other section to sort of deal with all of the other donors that, might not otherwise be captured. It's really hard because there are not as many gout sequences as I'd like, and other sequences that bind galacturonic acid, as I wish, to be able to build sort of a classifier for them. So I will say not yet, but at the very least, those sequences will be included on the next run. And it won't predict it as GAL-A or GLUC-A, but it will predict that the sequence belongs in an others category, and I will list what that category of donors will be. Yes.
Fascinating. And you talked about so much, you have hundreds of questions. I'm gonna narrow it down just to one. If we go back to the kinase, when you looked at the isoform,
Yes.
DCLK 1.1, 1.2, be sure I got terminology right. You're talking about one gene, alternative splice variant, so you got different proteins. Okay, you didn't talk about the biology, and that's kind of where I want to go at the very end.
Sure.
But you did something, I've heard some scientists use thermal stability as a first pass to try to get an idea of whether certain mutations might affect activity, and I've never understood that, because it doesn't seem to me there's necessarily a correlation, and your data clearly showed where sometimes you had more thermal stability and there was less activity, sometimes more. So my question to you, the first one, is what in your mind does, and that's a huge 15-degree increase in thermal stability.
Yeah.
So I'm going to get to that through biology. But in your mind, when you're looking at your variants and you're looking at thermal stability, what does that tell you? Because I don't understand.
So what that tells me is, so we're comparing thermostability with phosphoincorporation at the same time, so our activity assay. And I think those two in tandem really can tell us more about what exactly the kinase is doing. That is, if you have, for example, DCLK 1.1, if the tail is less, the kinase has activity, but the tail is less likely to actually bind the pocket. And so it's more free, it's not actually in the pocket. And so generally, we consider that less thermostable. Whereas a typical kinase, for example, will have that in an open conformation, we'll have that C-spine where there's pore packing, right? You have ATP bind that's what increases thermostability for that. And we know you bind ATP, you increase thermostability, and you increase overall stability for the enzyme. We're thinking that's essentially what's going on with DCLK 1.2 because it mimics the structure of ATP. It's binding that kinase, and that's what's contributing to that increase in thermostability. That's why also we see a complete inhibition on phosphate incorporation because that tail is essentially inhibiting ATP from binding.
Okay, so you need the total package to make any conclusion.
I think so. Yeah.
So just real quick to the biology.
Yes.
Because you didn't say what organisms you were looking at.
Sure.
Just from kinases in a biological control, you have these two different isoforms, one that's inhibited and one not.
Yeah.
And how is that control? Just quickly, what does that mean for the biology of the organism?
Sure. So first, we're using human kinase sequences, but we're doing in vitro experiments in E. coli. And in terms of the actual biology of how that's controlled, we note that both isoforms are expressed in the brain, but DCLK, but they have different expressions depending on the development stage. So DCLK 1.1 is expressed in adult human brain. DCLK 1.2 is significantly more expressed in fetal human brain. And so we think maybe it's as, once you're actually born, maybe DCLK 1.1, based on just literature reviews that I've seen and based on our data, maybe DCLK 1.1 becomes, sort of the dominant expressed isoform, and that helps sort of neural development during those later development stages. In terms of, I guess that, did that answer your question? I think.
Well, I guess the question is, why in the world do you express the 0.2 at all?
Yeah, and maybe it's difficult. I only know, that there are differences in the stage of development, but why that is, maybe there's some, I don't know much about neuroscience, so I won't comment too much about that. Yes.
But in the protein kinase realm, it's very common for protein kinases to be regulated by protein segments that either intercalate into the active site or interact adjacent to and impact substrate affinity. CAM kinase, PKA, all of those are examples of kinases that are inhibited, fully inactive in their ground state, and then through regulatory mechanisms, pull out an inhibitory segment and cause them to become activated.
Yes.
And there are a whole host of examples of that. So in the 1.2 variant, we've got a highly inhibited state. Are there any examples or any models by which one would anticipate regulating, pulling that inhibitory segment out in order to have that be an activated form?
Sure. So actually, that's a great question. And something I didn't mention was that DCLK is a calmodulin kinase, so the CAMK, and calmodulin kinases have that calmodulin domain that usually come in, pull out the tail, and help regulate activity. Now, a specific calmodulin has not been found for DCLKs, but that's likely, not enough, research has been done to figure out what substrate specifically is binding that. There's one potential one called HBCal1 that is expressed in the brain along with DCLK, but I don't think any experiments have been done to really evaluate the interactions between the two. Grace built an AlphaFold model of the two though, and it does bind right at exon 15 the tail. So that might be something shared between guys.
So the bottom line then is that the 1.1 version that isn't auto-inhibited versus the 1.2, and the potential swapping between those by splicing isn't necessarily the full story in regard to regulation.
Yes.
Because you also have the capability of regulating the auto-inhibit 1.2 by a calmodulin type of
Yes.
Very nice. I was wondering if the protein language model that you used in conjunction with AI could have gone and predicted the evolution of GTA family, or could it be used to have predicted the evolution of GTA family members, the Roskin fold, the end lobe extension, the C lobe?
That's a great question. And actually that falls into a lot of what Whalen had done before he graduated. He was looking into developing sequence embedding based trees. So this is an alternative to phylogenetics that is essentially looking at protein sequence alone and building a tree based on those sequence embeddings, those numerical representations of the sequences, to see how precisely they've evolved and where they fall in place in conjunction with each other. So yes, that is actually something you could look into. Whether people will trust that or not, I know that was a thing that Whalen struggled with in his reviews, if I'm correct, is how do you get people to trust it? And in what he decided was, it's better to say, embedding based trees are sort of an orthogonal approach to phylogeny. So if you use them in tandem together, they might reveal different aspects of evolution and tell you precisely how these enzymes have evolved. And so that is actually something we we started in the summer, but this Glidentify project ended up being sort of my main wheelhouse afterwards. But that is actually something we're looking into. Yes.
You had to skip very quickly through the concept of the ordered mechanism for glycosyltransferases, which is, something that's been discussed for a long time with the crystal structures of the donor binding and then sort of some generic loop closure that sets up the acceptor binding. Can you elaborate on what it is that your work contributed to that, or how it expanded on what had already been discussed?
So what my work essentially contributed is how the origin of those different modules that we see, the DXD motif, the origin of that. One of the things actually I didn't mention that was in my paper that's not in this presentation is we note that the topology of that phosphate binding cassette that contains that DXD motif that binds the metal cation, that topology is actually different in GTAs than it is for GTBs. GTBs more canonically resemble Rosman fold enzyme phosphate binding cassettes, whereas GPAs more closely resemble something called a pyrophosphorylase. And pyrophosphorylase, what they do are they take some uridine monophosphate sugar, and then they add a phosphate to it, creating a uridine diphosphate sugar. The exact, the exact donor substrate that glycosyltransferases use. And in fact, if you look at the operon structure of these, many pyrophosphorylases are now found, like I found just observationally next to glycosyltransferases. And so it's very possible that they may have evolved in tandem, and they might have a closer ancestral relationship than GTAs have with GTB enzymes. So that was one of the contributions, is that GTBs, we know, are metal independent versus GTAs, which are metal dependent. Pyrophosphorylase is also metal dependent. And of course, there are variations of that, GT116 that you found, right? You have, instead of the DXD motif, that DGK motif, the lysine acts instead as a potential positive cation for donor substrate rather than the actual metal cation. So these enzymes are not easy to just evolutionarily study because they're so divergent, and they're very artistic in the fact that they're more like abstract art than impressionist art, which is what I would consider like kinases, because they're very well conserved. And so they're very, very adaptable. And so that's essentially what one of the things that my paper had contributed is sort of looking at the origin of GTAs versus GTBs, specifically from these ancestral methods.
If I just heard you correctly, then your interpretation would be that GTAs are more closely evolutionary related to pyrophosphorylase than they are to GTB glycosyltransferases.
That is my working hypothesis, and that's what my data has shown. I don't think it'd be possible to prove that without some extensive phylogenetic analysis. However, an extensive phylogenetic analysis with all of those different enzyme families is very hard at this moment. But I think with AI and with these embedding trees that I was talking to Dr. West about, I think that might be a potential way to close the gap and figure out the relationships between different enzyme families, different enzyme superfamilies.
Okay, I think in the interest of time, we should probably stop here and thank Aarya for the committee members can stay and others can leave. So we'll send out an email as soon as the committee meets. You need some water or anything?
Yeah, I got water.
I'm wondering if
Great job.
Yeah,
Thank you so much.
Awesome.
I'm all in favor.
Thank you.
Thank you, there.